Okay, could somebody confirm you can hear me?
We can.
Thank you. Before we begin, are there any questions? Close that down. Let me share my screen. Should be seeing my screen now. All right, today is February 11th, and we should be discussing Chapter 5, Microbial Metabolism, hopefully ending this chapter, and beginning Chapter 6. Let me inform you that there will be a quiz this Wednesday, quiz 3, covering the end of chapter 3 and chapter 4 of the lecture, and labs 5 and 6. Any question about any of that? Blow it up so I can read it better. All right, let's go to chapter 5. I think we're further than that. Um, I moved where that is. Not sure where to find. There it is. Is this the last slide we talked about? Slide 45.
Based on my notes, the last slide was the nutritional classification of organisms.
Yeah, I think we were here. So that's slide 66. Let me get rid of this. This is something I didn't probably catch. That's it. All right. So last time we mentioned that you can digest lipids and proteins, but to do so, because lipids and proteins are big molecules, they cannot cross the cell membrane, that what we do as well as bacteria is um, we excrete enzymes outside the cell and break down the lipid and the proteins outside the cell, breaking them down into their monomers, which would be fatty acids for lipids and glycerol. And then for proteins, it's just breaking them down into amino acids. And then the smaller monomers can be brought across the cell membrane for further metabolism. All right, let's talk a little bit about the interrelationship of carbohydrate, lipid, and protein catabolism. Let me get that up there. Carbohydrates, lipids, and proteins, and really all food molecules, all organic molecules, can be a source of electrons and protons for aerobic respiration or for some bacteria for other forms of respiration. All enter glycolysis and have the Krebs cycle at various points. And you kind of already know this. If you eat too much carbohydrate, once you're done storing it as glycogen in your liver, where do we store it? If you eat too much carbohydrate, what's going to happen? You're not going to store it as carbohydrate. You're going to store it as
Fat.
fat. So we convert the carbohydrate, you already know this, to a lipid, and then you store it down as fat. Uh, the point of this slide is that carbohydrate, lipid, and protein catabolism happen when we eat these foods and they're usually broken down into glucose and then sent through aerobic respiration in the normal way. Let me blow the slide up a little bit. There are some exceptions. And that is a marathon runner can digest lipids directly and send the lipids into the what do you call it? The aerobic respiration, glycolysis, Krebs cycle, and electron transport chain, meaning they can digest lipids directly, but they don't do that until 45 minutes into their marathon run. So initially, they'll be digesting carbohydrates or converting the lipids into carbohydrates and then sending the carbohydrates in the normal way through aerobic respiration. Only 45 minutes into their run will they start digesting lipids directly and breaking the lipids into glycerol and fatty acids. Glycerol will actually enter the glycolysis midway through, and then the fatty acids will be converted into acetyl coenzyme A, meaning they're converted into two carbon chunks, converted into acetyl coenzyme A, and then enter the Krebs cycle at the start of the Krebs cycle. Most of the time, for most of us couch potatoes, what we will do is convert the lipids to glucose and then send glucose through the aerobic respiration the normal way. The same thing with protein. Most of us couch potatoes will convert proteins to glucose and then send glucose through aerobic respiration in the normal way. Any question about any of that? I haven't said how that happens, but we'll talk about that in a minute. Okay. Proteins are already mentioned, broken down into amino acids, and then the amino acids, depending on which amino acid they are, some of them can enter glycolysis, some of them can be converted into a two-carbon chunk and then converted into acetyl coenzyme A, entering the start of the Krebs cycle, and some of the amino acids are converted into a product and then converted into a late derivative of the Krebs cycle. For most of us couch potatoes, this will not directly enter the Krebs cycle. It'll actually be converted into glucose, and then glucose will come down the aerobic respiration the normal way. For carbohydrates, it's simple. Carbohydrates are either broken down into glucose or they're broken down into another sugar, and then that sugar is changed into glucose. Like fructose will be slowly converted into glucose by the liver, and then the glucose will be released into the blood for aerobic respiration the normal way. So for lipids, when couch potatoes digest a lipid, will break down into glycerol and fatty acids. Fatty acid will be coming into a two-carbon chunk, and then that two-carbon chunk will be converted into glucose. And then sent through aerobic respiration in the normal way. Like I said, you can directly burn lipids. A marathon runner will do that after 45 minutes in their run, in which case the fatty acid will be converted into acetyl coenzyme A and then enter the Krebs cycle. But the point of this is that carbohydrate, lipid, and protein catabolism are all interrelated. They all enter glycolysis of the Krebs cycle at various points, and they all can be digested. The complete metabolic oxidation of glucose and aerobic respiration is the most efficient. So aerobes grow a little faster than anaerobes under most situations. And like I said, using glucose and aerobic respiration is the most efficient. So those of us couch potatoes will convert those other food sources most of the time into glucose and then send glucose through aerobic respiration in the normal way. The energy that's obtained from aerobic respiration will be used to make ATP. The ATP will be used for various cellular processes, including active transport. You need one molecule of ATP to move one molecule across the cell membrane by an active transport protein. You can use ATP for movement, why my fingers are moving or why we walk, or why a bacteria beats its flagella. The ATP is giving the energy for that movement. And then we need ATP for the synthesis of new cellular components, which is anabolism. Most cellular ATP in cells is actually used in anabolism, including the biosynthesis of carbohydrates, lipids, and proteins. Any question about any of that? And as you know, lipids, proteins, and carbohydrates are three of the four biological molecules. Nucleic acid is the fourth. So anabolic and catabolic pathways are joined together in common intermediates. We call the pathway which can go both ways amphibolic. So a pathway that can function going this way, and then it can function going that way. These amphibolic pathways bridge the pathways involved in carbohydrate, lipid, and protein, and nucleic acid metabolism. So let me show you the bigger slide of this. Every pathway that has a double arrow means that normally the pathway goes this way, but it can go that way. So oxaloacetic acid can be converted into malic acid, etc. And then let's go ahead and start with this molecule. Suppose we have an amino acid we're digesting. We can then convert that one amino acid into oxaloacetic acid. We can then convert it this way into acetyl coenzyme A, a 2-carbon chunk. Convert acetyl coenzyme A into pyruvic acid, etc. Go this way, breaking the pyruvic acid eventually to glyceride 3-phosphate. And you see this arrow coming this way? Well, there's an arrow going that way too. So this is an amphibolic pathway. It's just splitting through ways, so they're not showing it with the double arrows. They're showing it with, I don't know, 3 arrows, meaning that this can be converted into that, and then this can be converted into glucose. And that's how we would convert this amino acid into glucose if you're a couch potato and you're digesting protein. We convert it into glucose, and then when we're ready, we use this glucose to send it through aerobic respiration in the normal way. The same thing happens with lipids. You eat fat broken down into glycerol and fatty acid. For us couch potatoes, that fatty acid will be converted to acetyl coenzyme A, and then that will be converted into glucose. For a marathon runner, they won't do that, well, after 45 minutes into the run, in which case the fatty acid will be converted into acetyl coenzyme A, and then the acetyl coenzyme A will enter the Krebs cycle the normal way, going through aerobic respiration beginning here. But they don't do that until 45 minutes into their run. I'm not sure, and probably it differs on which amino acid we're talking about when you digest protein. if you're like an Olympic athlete or a marathon runner, how do they digest that? When do they digest the protein directly? And I just don't know. With the marathon runner with the lipid, though, like I said, after 45 minutes into their run, they can digest the lipids. Anyways, these amphibolic pathways tell you that there are double arrows and the pathway can go either way. And that integrates the metabolism of carbohydrates, lipids, proteins, which I guess we don't have shown here, but there. Any question about any of that? So the amphibolic pathways bridge both catabolism, anabolism, and they integrate the metabolism of lipids, proteins, and carbohydrates. All right, let's talk briefly about photosynthesis. Photosynthesis is the conversion of light energy to chemical energy. The chemical energy may be used to convert atmospheric CO2 to glucose, and this is called carbon fixation, where green plants and other photosynthetic organisms can fix CO2 from the air or dissolved in the water and make glucose. This photosynthesis is performed in cyanobacteria, algae, and green plants. It's a mechanism for synthesizing organic compounds, and animals which cannot engage in photosynthesis require it to obtain their food. And you say, I eat beef. I don't eat plants. Well, where do you think the cow gets its food from? So all animals actually are dependent on photosynthesis. And there's only one ecosystem on the planet that isn't largely dependent on photosynthesis, the conversion of the energy in light to the energy in glucose by photosynthesis. And that's the deep sea vents, and they have chemosynthetic organisms which are supplying the energy for the system. It's not coming from light, it's actually coming from chemicals from the organic vent, and the chemosynthetic bacteria strip the electrons from the chemicals to get their energy needs. And then in their energy needs, they make glucose, fixing CO2 into glucose. And then that glucose becomes available to the other organisms in that ecosystem. But other than the deep sea vents, which do not rely on photosynthesis, the majority of life and the majority of the ecosystems on the planet are dependent on photosynthesis. Any question about any of that? All right, photosynthesis only occurs in photosynthetic organisms. These organisms contain a light trapping pigment. You actually know one of them, and that is chlorophyll. The light actually releases electrons from the chlorophyll, and then the light energy is converted to chemical energy via an electron transport chain. ATP and NADPH, that's similar to NADH, are generated, and they are often used to, meaning they're used up to make glucose from CO2 and water. And that step is called carbon fixation. There are two types of photosynthetic reactions. There's the cyclic photosynthesis and the non-cyclic photosynthesis. You probably learned the oxygenic non-cyclic photosynthesis, where 6 CO2, 12 water molecules in the presence of light form glucose, C6H12O6, 6 oxygen molecules, and 6 water. You can sometimes see the equation with the 6 water removed, and then they subtract 6 water molecules here. I prefer seeing this full equation with water on both sides, because when we look at where all of the 12 oxygen atoms came from, which make the 6 oxygen molecules of photosynthesis, aerobic photosynthesis, all of these oxygens come from the oxygen in water. So you do need to have 12 here to get 12 there to get the 12 here. Any question about any of that? Now, you do not need to know that there is another way that organisms can engage in non-cyclic photosynthesis. And instead of using water, they use sulfur, hydrogen sulfide, and then they make glucose and release sulfur. You don't need to know that equation. I'm not going to test you on it. Anyways, that's non-cyclic photosynthesis. What you've probably never come across yet until this lesson is that photosynthesis can also be cyclic photosynthesis, where instead of making ATP, which I don't have that on there, but this is making glucose here. And what you do is you make ATP and NADPH, and then you use up the ATP and the NADH to make the glucose. So it's a multi-step process. But that's non-cyclic photosynthesis. Well, cyclic photosynthesis, all you make is ATP. You use the electrons from chlorophyll, which are stripped from chlorophyll by the energy in sunlight. And then you use that electron to phosphorylate ADP and phosphate to make ATP. And all photosynthetic organisms can engage in either cyclic photosynthesis or non-cyclic photosynthesis. Like a green plant, if it needs ATP, it will engage in cyclic photosynthesis to make ATP. But if the green plant needs glucose, it'll engage in non-cyclic photosynthesis to make glucose. Any question about any of that? All right. Let me talk about the last slide of photosynthesis. I'm going to show you cyclic photosynthesis or cyclic photophosphorylation. Both terms are used. Photophosphorylation means we get phosphorylation of ATP right there. It's happening because of light photo. What happens, let me blow the slide up, is that light hits the chlorophyll, which is in the photosystem 1, and the light excites 2 electrons from the chlorophyll. Those electrons move out of the chlorophyll, moving to this protein in the membrane, moving to this protein in the membrane, moving to this protein in the membrane, and then moving back to this protein in the membrane. And then the electron ends up at chlorophyll, meaning the electron returns to where it started. And that's why it's called cyclic photophosphorylation. Oops. The electron moves in a circle, returning to where it started from. Along the way, the electron at this protein gives this protein the energy it needs to pump hydrogen ions from this side of the membrane to that side of the membrane. The hydrogen ions create a twofold gradient. There's more hydrogen on this side than that side. There's also more positive charges on this side than on this side. So these hydrogen ions will want to move by the laws of thermodynamics to this side of the membrane. They can only pass through the lipid bilayer at this channel protein. And when the hydrogen ion moves through this channel protein, it gives this protein the energy it needs to phosphorylate ADP and phosphate to make ATP. So this channel protein is actually ATP synthase. That should be familiar to you, hopefully. It's very similar to what's going on in the electron transport chain of aerobic respiration, where the electrons are carried to this protein in the membrane, and then they're transferred to this protein in the membrane, to that, to that, to that. And then in aerobic respiration, the electrons go to oxygen. Along the way, the electrons give certain of the proteins the energy they need to transfer hydrogen from this side of the membrane, I should say hydrogen ion from this side of the membrane to that side of the membrane. And then the hydrogen ions are used as potential energy by this channel protein. And when the hydrogen ions move through this channel protein, they give this protein the the energy it needs to phosphorylate ADP and phosphate to make ATP. Any question about any of that? I'm sorry, I'm a little out of place here. Or I should have done, just go to the last slide. That would be easier. Oh yeah. Where does this happen in a eukaryote cell? This happens in the chloroplast. So this will be inside the matrix of the chloroplast, and this will be outside the the inner membrane of the chloroplast. In a chloroplast, we have an outer membrane, and that will keep the hydrogen ions inside the chloroplast, between the inner membrane of the chloroplast and the outer membrane of the chloroplast. In a prokaryote, which do not have chloroplast, these membranes in the, these proteins in the membrane are in the cell membrane, and this will be inside the cell or the cytoplasm, and the hydrogen ions are pumped outside the cell membrane. So this will most likely be the cell wall, and then the hydrogen ions come through the membrane at this protein, and that's what causes ATP to be made in cyclic phosphorylation. Any question about any of that? All right. The last slide of this lesson is looking at the requirements of ATP production. The energy source can vary. That's the electron donor. We talked about with photosynthesis, the electrons or the energy source comes from the light in the sun, which then hits the chlorophyll, and then the chlorophyll supplies the electron, donates the electrons. The electron then moves through electron carriers. It's actually NADP plus in green plants, but in animal cells, the electron comes from glucose. But for chemosynthetic bacteria, it can come from an element like sulfur or ammonia or hydrogen gas. The electron will be stripped from those chemicals and then go through the electron carriers, which most of the time will be NAD plus and FAD, same as in aerobic respiration. And then the electron acceptor can vary. It's not shown on here, but the electron acceptor for cyclic photosynthesis will be the same molecule that donated the electron, and that would be the chlorophyll. For aerobic respiration, the final electron acceptor is oxygen. But for anaerobic respiration, it'll be some other chemical other than oxygen. It can be nitrite or nitrate. It can be sulfate, just depending on which form of anaerobic respiration we're using. It'll have a different final electron acceptor. And then for fermentation, the final electron acceptor will be an organic chemical compound. So the electron source, energy source can vary, and the electron acceptor can also vary. Any question about the requirements of ATP production? Or any questions about Chapter 5 in general? All right, let's close that down and begin Chapter 6. Chapter 6, Microbial Growth. This slide is showing you the major goals and a rough outline. So always know the terms. What's a psychrophile, a psychotroph, a mesophile, thermophile, hypothermophile? What's an acetophile? What's a halophile? What is an aerotolerant anaerobe? A facultative anaerobe? What is binary fission? What are reproductive spores? And what are endospores? Remember, to know all of the goals of this lesson, you need to learn the objectives, and that you can find by looking at the study guide for this chapter. At the start of the study guide will be the objectives, and all of the quiz questions will come from the objectives. Any question about any of that? All right. 2, understand the various requirements that cells have for growth. 3, understand the 4 phases of growth. And 4, compare the different methods we have of measuring microbial growth. When we're talking about microbial growth, we are referring to the increase in the number of cells, not the cell size. I once taught middle school students, and when we talked about them growing, we weren't talking about them increasing in number. Any questions about that? Let's watch a little short video on bacterial growth.
Bacteria reproduce very simply and rapidly by doubling their contents and splitting in two. Just one bacterium, dividing every 20 minutes, could produce nearly 5,000 billion bacteria in one day.
Any question about that? All right. There are requirements for microbial growth, and you could say that for all life, but here we're only talking about the requirements for microbial growth. There are physical requirements. That includes things like temperature, pH, osmotic pressure, salinity, and there are chemical requirements. All living cells have to have a source of carbon, a source of nitrogen, a source of sulfur, source of phosphorus, source of trace elements, oxygen if they are requiring oxygen. If they are not requiring oxygen, they may require oxygen to be absent. And if they require it, they may require an organic growth factor. Each species, when we're talking about temperature, has its minimum temperature that it can reproduce at. It's not maximum temperature that it can reproduce at, and its optimum temperature for growing or reproducing at its fastest rate. Microorganisms are classified into groups on the basis of their preferred temperature range. The psychrophiles are organisms that grow best around 10 degrees Celsius. That's the temperature of your refrigerator. The psychotrophs grow best around room temperature. The mesophiles grow best around 37 degrees Celsius. The thermophiles grow best around 60 degrees or a little above that, 62 degrees Celsius. And the hypothermophiles grow best around 95 degrees Celsius. Recall that 100 degrees Celsius is the temperature that water boils at, sorry. Now realize that not all ranges are rigidly defined. The different organisms can even have a different optimum, like Micrococcus luteus is considered a mesophile, but it actually doesn't grow best at 37 degrees Celsius. It grows best around 30 degrees Celsius. And the reason is Micrococcus luteus tends to grow on our skin, and so its optimal growth rate is actually at 30 degrees Celsius, which is the temperature of our skin. Some organisms like E. coli, which are inside our gut, grow best or have an optimal temperature at 37 degrees Celsius. The point is, they can have different mean optimals and then different ranges for what their minimum and what their maximum is. So they're not rigidly defined. We just say anything that has an optimum around 37 degrees as a mesophile. But if it's around 25 degrees, we call it a psychotroph. Any questions about any of that? All right. Talking about the mesophiles, the psychotroph, and the psychrophiles grow best at a temperature that is the temperature of your refrigerator, and given enough time, they will grow and then cause spoilage of the food. The psychophiles need about a week before their numbers are large enough that they can spoil the food. And that's why food, unless it has a preservative in it, like a lot of sugar or a lot of acid or something like that, a lot of saline, that you should throw them out after a week in the refrigerator. Meaning the psychrophiles can grow in the refrigerator and cause spoilage of the food. If there's preservatives in it, like either a lot of sugar, or a lot of salt, or a lot of acids, like ketchup, which has a lot of all three of those, you can keep it in the refrigerator for much longer than a week. How long can you keep it? I'm not sure. But my mother and I had a bottle of ketchup that was several years old. So let's say 2 to 5 years old. It wasn't going bad. And we finally gave it to my brother because he uses ketchup all the time on almost everything. And my mother and I didn't use ketchup very much, and that's why we had a bottle of ketchup that was several years old. Any question about that? I could tell you I've got a bottle of ketchup that is at least from 2018. So whatever that is that 7 years now? Boy, that's getting old. And I'm still using it, still eating it, and slowly, and it hasn't gone bad. So if you have a lot of preservatives, like I said, it can last much longer than a week. We were talking about food spoilage. Let's finish that discussion, and that is food can spoil whenever it is in the temperature range between 60 degrees Fahrenheit to around 130, and that would be something like 15 or 16 degrees Celsius to 51 or 52 degrees Celsius. At this temperature, bacteria can grow rapidly. Some of them can grow rapidly and then produce a toxin, so that if the food is kept at this temperature for more than 4 hours, the food should probably be thrown away because the bacteria can grow, produce a toxin, and then with the toxin, the food will look and taste fine, but then the toxin will do you in. Many bacteria can grow around that range, but slower. Like below 62 degrees, we can have the bacteria growing between 52 to 62. Bacteria can grow slowly. Or down to really about 5 degrees Celsius, up to about 15 degrees Celsius. The bacteria can survive, some can grow slowly. And so the bacteria can spoil the food there too. As I stated, the psychotrophs can grow in the food, and they will grow in the food growing in your refrigerator. So after a week, if there's no preservative, the food should be thrown away. Of course, above that temperature, we don't have many bacteria growing that can spoil food. Like the hypothermophiles could grow in this temperature range, but for some reason we don't have any hypothermophile known that causes human disease. And for some reason, the hypothermophiles don't tend to spoil the food also. And then below freezing, we don't have bacteria growing. That means the bacteria might be alive, but they're frozen and they're just sort of held in stasis. There's very little, if any, metabolic activity in a frozen cell. But then the bacteria, if they're thawed out, about 50% of the cells can survive freezing and then thawing. But the bacteria cannot spoil the food if it's frozen because the bacteria are not metabolically active. So when you refrigerate your food, you should split it into small batches. If you have it in a large, very large batch, the food will cool, but only slowly, and the food can stay in the danger zone for greater than 4 hours. 1, 2, 3, 4. Yeah, before there. So that's over 4 hours. And when it's over 4 hours in this danger zone, the bacteria can grow and then produce an endotoxin, causing the food to spoil. So always refrigerate in a small batch so that the food can cool quickly, obviously less than 4 hours. Here it looks like it's actually a little bit less than 1 hour. And then the food can go through this danger zone quickly and then cool to a temperature where the food is unlikely to cause problems. Any question about any of that? All right. Another requirement that cells have for growth is the pH. most bacteria grow between pH 6 and pH 7, specifically pH 6.5 and 7.5. However, there are some bacteria that can grow outside that pH range. In fact, when we're growing cultures in the lab, most of the bacteria, when they're in the culture, will produce acids, and these acid products can then inhibit the growth. To minimize this, we typically add pH buffers to the media to buffer the pH, and then the acid products that are made will be buffered so the pH of the media will slow and will not become acidic until after many hours or days. In which case, when it becomes too acidic, the microbe cannot grow. But we put the buffer in there so that the microbe can continue growing for much longer than if the pH buffer was not in the media. Any questions about any of that? All right, molds and yeast do not grow well around pH 7. I mean, they can grow at pH 7, but they actually grow best between pH 5 and 6. So that's somewhat more acidic than bacteria. It's a slight problem if you have a mild acid, like you put cucumbers and vinegar in your refrigerator or you pickled your beets. If it's not acidic enough, the molds and the yeast can grow. And they would then cause spoilage of the food. Acidophiles are microbes that can grow best in an acidic environment, meaning one less than pH 7. And to usually get a test of an acidophile, you would grow the bacteria in different pHs, something like pH 1, pH 2, pH 3, pH 4, pH 5, pH 6, if you suspect it's an acidophile. If it's an alkaline, you'd have to go higher than pH 7. And you wouldn't go less than pH 7. Acidophiles are organisms that grow best in acidic environments, such as volcanic environments where the pH will be pH 0 to pH 3. Where is a place in or on your body where you might find acidophiles, something growing in a extreme pH, like pH 2 or less? Anyone know?
Could it be the stomach?
Yeah, your stomach. Your stomach will tend to have acidophiles. It obviously will depend on what acids your stomach is making and how long after a meal and what you've eaten as well. Because if you eat food with buffers in it, like a lot of vegetables, they tend to buffer the pH, and then your stomach will not become as acidic. The stomach acids go down to pH 2 or maybe even pH 1.5. They don't go down below pH 1 though. Stomach isn't that acidic, but the most acidic thing you can come across that you are familiar with would be the acid in your car battery. And that is very close to pH 0. So acidophiles are organisms that like to grow in an acidic environment. You can also have the requirement of osmotic pressure. Microbes always require water. Without water being present, the microbes cannot engage in metabolism. And generally, if that happens, the cell will begin to die, either of thirst or of a lack of energy, meaning you need water around for metabolism, and One of the forms of metabolism is respiration, making ATP. High osmotic pressures can remove the necessary water from a cell, where a hypertonic environment where there's more solute concentration outside the cell than inside, that will pull the water out of the cell. Let me blow this up. So water will leave the cell. That will cause the plasma membrane to pull away from the cell wall. You can actually see the plasma membrane shrinking, and then it will form into a, forgot what that's called, a compaction of the cell membrane with water leaving the cell. For many cells, this will be toxic. Like for E. coli, this would be toxic. So E. coli, once it falls into a hypertonic solution, the water will leave the cell, and that will kill E. coli. Staphylococcus aureus is not killed as easily. That's why Staphylococcus aureus can live on the skin. Even when the soil, as the skin is at its most salty, like after you sweat it a lot and then you evaporate the sweat, the skin will be approaching 20% sodium chloride. For many bacteria, that will be toxic as water leaves the cytoplasm. But for Staphylococcus aureus, it can survive that. So high osmotic pressure can be used to preserve food. We've been doing that for centuries, where when we make jellies and jams, for example, the high sugar content is used as a preservative. Most other bacteria cannot grow in that. Honey is also using a high sugar content, both glucose and fructose, to preserve the honey. And then when the bacteria fall on the honey, it will go through plasmolysis, where the plasma, the cell membrane, the cell plasma pulls away from the cell wall and then moves into the cell, inside the cell to the center of the cell, and will shed water. Another example besides honey is molasses, where the high sugar content of the molasses prevents the bacteria from growing. And I mentioned, I think, that my mother had a bottle of molasses that we figured was at least 30 years old. Hadn't been used for 10 years, meaning it was 20 years old when I used it to give my father some molasses candy. But that was the last time it was used. So for 10 years, it wasn't being used, and my mother finally decided to throw the bottle of molasses away, which was fine because she didn't use it and I didn't use it. Didn't make any more molasses candy for my father because shortly after I made it, a few years, he died. Halophiles, you don't need to know that last part I just gave you for information. Halophiles are organisms that like growing in a high salt condition. We have two types of halophiles: an obligate halophile. These are organisms that require high salt concentrations in order to grow, like the organisms growing in the Dead Sea between Jordan and Palestine. Those will be obligate halophiles, and they grow only in the case of high salt concentrations. And if you don't know, the Dead Sea is got salt crystals in it, meaning there's so much sodium chloride in the water of the Dead Sea, the salt will start forming a inclusion body. So much will accumulate that it just starts precipitating out of solution in the cell. Anyways, that's the obligate halophile. They require high salt concentrations to grow. And then if you remove the high salt concentration, the cell will either go into stasis or it will die. A facultative halophile, on the other hand, can handle the high salt concentration, but it does not require it. Like Staphylococcus aureus, can grow on a normal nutrient agar plate where the sodium chloride concentration is 2% or less, but the Staphylococcus aureus can grow on MSA where the sodium chloride is 7% or higher. And it can grow on your skin, which can grow up to 70% sodium chloride. Did I say that right? The skin can go up to 20% sodium chloride. So the facultative halophiles can live either in high salt concentration, but they can also grow and divide at, I don't know, low salt concentration. And low salt would be something isotonic or similar to that. Doesn't have to be 0 salt. In a hypotonic solution, low osmotic pressure, having more distilled water outside the cell, water will enter the cell by osmosis, causing the cell membrane to expand. If the cell has a cell wall, the cell membrane will expand all the way up into the cell wall. The cell wall then protects the cell membrane, and it will not expand further. There's no more room for the water inside the cell. So the cell membrane cannot expand any further. No further water can move into the cell because there's no room for it. All the space has been already taken up by water. Hypotonic solution, meaning low osmotic pressure, lots of water outside the cell. Water enters the cell, and that may cause the cell to lyse. As the cytoplasm expands and expands and expands, up until the cytoplasm will burst. A cell wall will prevent that, and then the cell membrane can only expand up into the cell wall. It can't go any further, and that will limit the water's movement into the cell because there's no room for the water to move into the cell. Any question about any of that? All right. Cells also have a carbon requirement. Carbon is typically half of the dry weight of a cell, meaning if you take a cell and remove all the water, half of that weight of the cell will be carbon. Carbon is used as a structural organic molecule. You can find it in the cell membrane. You can find it attached to generally proteins circulating in the blood. Carbon is an important molecule for generating energy. I mean, you get carbon from a molecule which gives you the energy. And most of the time that would be glucose, at least for humans, but it can be another food molecule. So we do need carbon. The heterotrophs are an organism that uses an organic carbon source. So humans get their carbon from digesting glucose or some other food product. And then an autotroph is an organism which can use carbon directly from CO2 in the air or the water. Any question about any of that? All right, nitrogen, sulfur, and phosphorus are other chemicals that are required for life. Nitrogen, about 14% of the dry weight of a cell is from nitrogen. You can find nitrogen in amino acids, proteins, DNA, RNA, ATP. Many bacteria can obtain their source of nitrogen from decomposition of a amino acid or decomposition of a protein. Many bacteria can use nitrogen, though, from either ammonium or a nitrate. So the nitrogen can come from protein, but it can come from either ammonium, or nitrate, which oftentimes is supplied directly into the media. A few bacteria can use nitrogen gas in the air, and these we call nitrogen fixing bacteria because they're pulling nitrogen out of the air and then converting it into an organic form of nitrogen, meaning if it's a plant pulling nitrogen out of the air, then nitrogen typically is held by some structure in the plant. It might be the root, it could be something else. Sulfur is also a chemical that is required for life. Most organisms get their sulfur from breaking down proteins into amino acids. The amino acid Feely? Is that the dwarf who did it in Lord of the Rings? Some bacteria can get their sulfur from breaking down amino acids, meaning breaking down proteins. Some can get it from thiamine or biotin. Sulfur is required for DNA and RNA synthesis, as well as obviously certain synthesis of amino acids. Thiamine is a B vitamin, including sulfur. Biotin also. Some organisms can break the sulfur down from protein, but many bacteria can use sulfate or hydrogen sulfide gas as a sulfur source. Hydrogen sulfide gas would be toxic to us, so we can't use it. Sulfate is something that we as humans cannot use, but green plants can use sulfate. And you can look on the ingredients of a fertilizer, and one of the first will be sulfate. The first usually is either the superhero media. Can't remember the name of that superhero though. Oh well, the point is that bacteria can get sulfur from protein or amino acid decomposition. Many bacteria can get sulfur from sulfate or hydrogen gas, which would be supplied in the media. Phosphorus, another chemical requirement. All living cells must have a phosphorus source. Phosphorus is required for DNA and RNA synthesis. It's also required for ATP synthesis. And then phosphorus is found in the membrane of our cells. The phospholipid comes about because the glycerol, instead of binding to two molecules of sorry, I lost what I was trying to say. What the heck is the name of that? Phosphorus. It's the main molecule in the cell membrane or any membrane because it's in the phospholipid. Most organisms can use as their source of phosphorus phosphate, which you can find in bone and a little bit in meat, but mostly in bone and things like that, have a lot of phosphorus. If you're consuming DNA from the cell, meaning you have well, let's say an apple. It has a apple cell. You can digest that cell, and inside the nucleus of that cell would be DNA. And we can digest that DNA. We do have the enzymes to digest DNA. Anyways, phosphorus comes from phosphate. You can find phosphate naturally in milk. All right, any questions about any of that? Um, finish this slide in time, and maybe we can finish the next one in time too. Yeah. All organisms require trace elements. These are inorganic elements required by microbes in small amounts, required for the microbe to grow and divide. Perhaps the element takes place part in an enzymatic reaction. Anyways, you need these elements in trace amounts while they're called trace elements. They're often required as enzyme cofactors. An example of a trace element would be iron, copper, and zinc. Generally speaking, most organisms get the iron, the copper, and the zinc they need from their environment, meaning humans get enough copper and zinc generally from the food they eat or the liquids they drink. And that's how most organisms get their trace elements, just from their environment that they're in. Any question about any of that? Organic growth factors are organic compounds obtained from the environment that the organism cannot synthesize itself. So to get the organisms to grow, you have to add the organic growth factor. Humans do have organic growth factors that they have to get from their diet because they cannot make the following molecules on their own. So they have to get it from their diet. And if they don't get it, they will eventually become sick and eventually die. One example of an organic growth factor for humans would be vitamin C. We cannot make this molecule. We have to get it from our environment. And if we don't get enough, we will become sick. And eventually, if you're really shy on vitamin C, you would die. Certain amino acids are organic growth factors, and these are called essential amino acids. Essential amino acids are the amino acids that you must get in your diet. Because if you don't get them, you will not have that amino acid to add to a growing protein when the protein is trying to grow. Certain fatty acids are also essential, meaning humans must get it in their diet, and these we call essential fatty acids. Some bacteria can have certain amino acids or certain fatty acids required, meaning they has to be present in the media or they will not grow. We talked about Streptococcus pyogenes having, what is it, factor X and factor V from the blood. And you must supply those factors to the media or Streptococcus pyogenes will not grow. And typically how we get Streptococcus pyogenes to grow in the lab is we just grow it on blood agar, and then the plate has enough of the bacteria on the seat. But if you sit down, you pick up the bacteria, and then the bacteria can migrate to the anus and then go inside your anus, providing, I guess, what you need, whatever that thing for. The purines and the pyrimidines can be an organic growth factor for some bacteria, where they must get the purine or the pyrimidine from their environment because they cannot synthesize it on their own. Any question about any of that? If not, I think I'm going to stop here. We'll stop a little early. This slide is going to be 3 slides. And I don't want to talk about oxygen yet. Any questions? If not, for those of you in the 6:30 lab, I'll see you at 6:30. Those of you in the lecture only, I'll see you in the next lecture or the next lab at 2:30.
I have a question.
Yeah,
Do you know when the lecture for last Thursday will be up on YouTube? I noticed it wasn't there.
last Thursday, well, we finished, if you're talking about lecture 5, I finished
Yes.
it just today, and that's why it wasn't up.
Oh, okay. Okay, thank you.
Let me see. Lecture 5 is not on the next quiz,
Okay.
so I'll put it up later this week.
Okay, thank you so much.
Right. All right, any other questions? If not, I'll see you.